The overall goal of the following experiment is to microfabricate devices used to mechanically stimulate cells in 2- and 3-dimensional culture environments. This is achieved by using a multi-layer sandwich molding and stacking technique to produce an array of vertically actuated microposts. As a second step, the vertically actuated micropost array is modified to stretch a culture film, which applies well-controlled strains to cells cultured on the film. Alternatively, the array can be modified to compress micro-patterned biomaterial constructs in order to apply mechanical stimulation to cells cultured in an array of 3-dimensional hydrogels. Results are obtained that show the simultaneous application of a range of 2-dimensional equibiaxial and 3-dimensional compression strains across the array based on image analysis of fluorescent fiduciary markers.
Hi, I'm Chris Moraes from the labs of Craig Simmons and Yu Sun in the Department of Mechanical and Industrial Engineering and the Institute of Biomaterials and Biomedical Engineering at the University of Toronto. Today we'll show you a procedure to fabricate microdevices used to mechanically stimulate cells. We use this procedure in our lab to study how cells respond to a variety of mechanical forces in vitro culture. So let's get started.
The process of sandwich mold fabrication includes placing an overhead transparency onto uncured polydimethylsiloxane, or PDMS, on an SU8 master. The sandwich is placed in a foam, glass and metal stack and cured in an oven under compression. Finally, the stack is disassembled and the transparency peeled away.